We don't want just anything happened without enzymes. Enzymes, already we talked about it. Enzymes, they could be embedded in the phospholipid bilayer, but also the enzymes, they could be attached outside of the membrane or even be inside of the cytosol. But in this case, because we're talking about the cell membrane, we will talk about the enzymes which are embedded in the phospholipid bilayer. And again, with the help of those enzymes, the chemical reactions will go, and a lot of this reaction are maintaining the homeostasis. Now, the proteins which are embedded in the membrane, plasma membrane, structure, they provide structural support. Remember, we are talking about cytoskeleton. This is the sites where the these fibers attached to create it. They could be both on outside, on inside, but mainly it's on the inside to provide the anchoring part to the this cytoskeleton. And the last one, but not the least, is the proteins which are anchoring cells to each other. So, for example, here there are two adjacent cells. They are tied together with the special proteins, and they also can provide the communication between the cells. Sometimes they could have a little channels or pores within these proteins, and of course, will be easier for these cells to communicate. So this is the mosaic representation of different proteins which creating this diversity on the plasma membrane, which is not just separating two liquid parts, but also allowing the communication, the structure, the processes are going through. The properties of the membrane, what's happening, and we will have some experiments done with the special membrane, but also we have different kinds of the transport. We talked about the concentration and gradient already. There's three variables to determine how this substance is going to move across the plasma membrane or being transport. We have two parts. We have passive and active transport. Active transport requires some energy. Remember these adenosine triphosphate molecules which were storing the energy which were acquired from the cleavage of the molecule of the glucose, which we acquired through our food. So this energy is used to move something or transport something through the membrane, and it's called active. And if we have transport which is passive, it does not require any energy. This is two the kind of differences between active and passive transport. But anyhow, how the substance is moving across the plasma membrane. It depends on the type of the substance. And also it depends on the membrane ability to let the substance through, what's called permeability. Because it could have, for example, if we have a small kind of filter and the openings are not big enough, the substance will stay on one side. It all depends on the quality of the membrane. And also we have the concentration of the substance. How high is the concentration on one side compared to the concentration on the other side? You know that by simple diffusion, when we have a lot of high concentration on one side and lower concentration on the other side, they will just like to go there, they will like to occupy even amount of substance there. And there's a simple representation how it works. You probably did it in your life several times, maybe not even thinking about it, like when you have the high concentration, for example, if you have the tea is too strong and you decide to make it slightly weaker, you will just add plain water, like boiling water to it, and the concentration will be less. But in the beginning, when you have high concentration of the molecules in one place and low concentration in another place, you can see how they start to move from one place to another. How it's happening in the cell? First of all, by simple diffusion. Simple diffusion doesn't require any special efforts from the cell. The molecules are so small, they could just diffuse through the membrane without any energy or without any efforts. They are going from the place with the high concentration into the place with low concentration. That's how the gases are moving in and out cells, like oxygen and carbon dioxide. They just diffuse. The facilitated diffusion requires, again, it's still diffusion, but in order to move through the membrane, these molecules or ions, for example, we have sodium and ions moving through, we have to have special openings there. So channels, they could be open constantly, and again, the diffusion happens through these channels. Another openings we will call the polar ionic compounds, which are moved with the special carriers or protein carriers. Remember, I was talking about the protein, which will attach itself to the substance and kind of help it through the membrane. This is another representation. I have to talk about the special process which called osmosis. Okay. The osmosis is the passive process, and the process which is happening in the environment when we have the membrane. For example, we have the membrane here. This is phospholipid membrane of the cell, and we have cytosol inside of the cell, and we have extracellular fluid here. And by the means of the osmosis, solvent, remember solvent and solute? Does everyone remember what it is? So solvent is what is dissolving something, and the solute is the usually used to be more or less solid structure, solid substance, which is dissolved by water in our case. So water moves from the area, from the low concentration of the solute, more water molecules across the membrane to the area of the high concentration of the solute, less water molecules, because the solute itself cannot move, only water. The solvent will move. The water will move from one side to another side. So osmosis and diffusion, they are very different forces for three reasons. The osmosis requires presence of the membrane, diffusion does not require. Remember the cup with the tea, we add more hot water, there is absolutely no membrane. Osmosis is reversible, so we go back and forth when we change the concentration on each side of the membrane. It's possible to move that water or solvent from one side to another, depending on the concentration of the solute. Diffusion, we cannot if we mixed lemonade there, that's it. That's what happened. We cannot move it back. Solute movement in diffusion can be predicted by the laws, and solvent movement in osmosis, we cannot predict that. This is a little bit too much of the physics and chemistry. Why is all this stuff important? You would ask me. It is very important, but because we are dealing with the liquid parts of our body, well, we're dealing with the extracellular fluid and intracellular fluid. How we can see it on practice. For example, yes, there will be a movie on your canvas, which is dedicated to the lab, and you would see we usually did it in the lab when we took a little bit of blood, a drop of blood, and we placed the red blood cells in the different in the solutions of the different concentrations, isotonic, hypotonic, or hypertonic solutions. And because the red blood cells, like any other cells, they do have the cellular membrane, we saw the effects of the osmosis, or tonicity is the way to compare osmotic pressure gradients between two solutions. We would see what will happen to the red blood cells if they are placed into the different solutions. For example, if the right there on the top of this slide, we can see the normal red blood cells placed in the isotonic solution. It means that the concentration of the solute is equal on both sides of the membrane. So there's an equal amount of the molecules there, and the shape of the cell is preserved. That's why when someone is going to the emergency room and they say, okay, this person is dehydrated, they usually give them a bag of isotonic solution to replenish their blood volume. It's very important that it's not hypertonic or hypotonic. Hypertonic will be plain water. Hypertonic solution will have a lot of salt or other solvents dissolved in it, solids dissolve in it. And what could happen to the cells if the solution is hypertonic there? We will have a lot of molecules outside, and the solvent or plasma or intracellular fluid will flow through this membrane into extracellular fluid. And since the shape and volume of the cell is predetermined, what will happen? The cell will basically shrivel. And of course, it will lose its function and it will be crenated. The crenation will occur. If we have the less concentration on outside of the cell, for example, we put just plain water for some reason, someone made a mistake and they injected plain, distilled water in the bloodstream, what will happen? Immediately we have very low concentration and extracellular fluid. And this fluid will rush through this membrane inside of the cell because we have concentration is much higher here. Again, the shape, the volume of the cell is predetermined, and it will absorb as much water as it can till it completely bursts and the membrane will be broken. And of course, a cell will lose its function, its structure. And if it's done on a bigger scale, of course, it can give us a hemolysis. All the blood cells would be destroyed. Of course, this is devastating things of the human body. That's why the only isotonic solution is used for intravenously to inject a person if they are missing the blood volume or any kind of other signs of dehydration. Do you have any questions about this process? It's very important since you're going to be healthcare practitioners. You have to understand what's going on. Yes, no feedback? Is everyone awake?
Yes.
Yes, you're awake. Good.
Yep.
The rest are asleep. Okay. Do you need a break, or we keep going? We shall keep going.
I have a question actually.
Yes. Yes.
What I see on the left side of the plasma membrane, it says cytosol.
Cytosol? Mhm.
Yeah, and on the right side it says ECF. What does ECF stand for?
Extracellular fluid. Cytosol, it will be intracellular, inside of the cell, and extracellular fluid is outside of the cell.
Oh, okay.
Okay. Okay. Any other questions, madam?
So how are we gonna memorize all this? Because there is a lot.
How you going to memorize? By looking at the pictures and answering the questions.
So the question and the quiz, it will have.
Yes, you will have the questions about this. Do I need to repeat about the concentration? If I need to repeat, I will repeat it.
Yes, please.
Okay, we have 3 variations, right? The concentration could be equal on the both sides of the membrane, right? For example, there's 50% and 50%. Since the concentration is equal, water will go back and forth without changing the shape of the cell.
Okay.
Nothing happens. We just, if we need to replenish the volume of the blood, we cannot do without put in like extra blood, for example, the blood is not available, but the volume of the blood is low in the person, they dehydrated, they lost liquids for some reason. Immediately we can give them this isotonic solution and nothing will happen, just we have more fluids there, so we replenish the fluids. But if we have the concentration, extracellular ECF, will have the constant concentration of let's say solution is much higher than inside of the cell. For example, it's seawater, very salty, or some high percentage of the glucose, like for example 10%. What will happen? We have more water within the cell, right? And it will go through the membrane into extracellular fluid, because it will try to dissolve. Only water, only liquid part can move through. The cell itself will shrink because it will lose the volume of the cytosol or the fluid within the cell, because all the liquid will go outside, try to dissolve this part. That's physics. That's how it's doing. It's moving from the high concentration into low concentration. If we have opposite situation and we have very diluted solution outside of the cell, for example, distilled water, absolutely no molecules there. Again, the liquid will try to go from outside into the more concentrated environment within the cell. And because the cell is basically a bag, and it will accommodate as much of the extra water as it can, but it cannot stretch forever and it will burst. It will swell first and then burst, destroy. Is that clear?
Thank you.
Okay, so there's 3 cases. Iso means the same, hyper means too much, and hyper means too low. But please, let's continue. Let's don't dwell here. That's the Okay, the next part, we already talked about the diffusion. It's like free movement through the membrane. Then we talked about the osmosis. It's only moving of the solvent or liquid parts through membrane from area of the low concentration of the this liquid to the area of the high solute concentration. Active transport, remember the active transport, it requires extra energy, or before the we talked about the diffusion and osmosis, it was passive transport, but active transport, it requires energy, it requires ATPs, because we are going to move these solutes against the concentration. We will move it from the low part to the high, and that requires extra effort. Before that, it was flowing freely, but now, for some reasons, we need these molecules inside of the cell versus outside. Even we're kind of accumulating more goods from outside. We're bringing more and more and more. We have two parts. We have primary and secondary active transport, and they are still using the same plasma membrane carriers proteins, which are specially designed to carry something through the membrane with the help of the ATP, adenosine triphosphate. They attach the required substance and they move it into the cell. So we call them pumps. There's three types of pumps. So we have, again, three types: uniport, symport, and antiport. Again, uniport, uni means what? Uni means one, right? So uni transport just one single substance through the membrane in one direction. Very simple, just one way, one substance. But we're trying to conserve energy. We're trying to use kind of piggyback on the transport, because we want to save some of these ATPs for, they're not abundant, so we're saving an energy. That's why our body designed some other means to do transportation, because we don't just need to bring stuff in or out, but at the same time we can move two or more substances through the membrane in the same directions, kind of allied. Something is moving hand in hand, and we are going. And antiport, anti means opposite directions. If we bring some nutrients in, we can take the waste out while the door is open. So this is the opposite direction. And again, uniport, one substance, one direction. Symport, two substances, one direction, the same direction, in and out, doesn't matter. And antiport, two substances going opposite directions. How would you remember it? I would suggest you to try to remember what the words mean, like uni, sim, anti. This is the part of the learning medical language. It's again, this is Latin, a little bit of Latin for those of you who speak Spanish as another language. Probably a little bit more easier, because Latin has a lot of in common with the Spanish, more than English. But for the rest of you, you just have to memorize it. I don't see another way about it. All right, here we have the examples of the secondary active transport. Again, we see the molecule of ATP here attached to the pump, to the structure, the protein structure with its pumping. In this case, we have potassium and we have sodium pumping through. And we're using all these ATPs in a storage. But again, we are doing ATPs, they are used to create and maintain concentration gradient of the one substance. So while it's open, the other substance is going through, and we can use the same ATPs. Okay, secondary active transport, we see on the right, the substance moving across the plasma membrane down concentration gradient and moves the another substance again. So remember, we bring one in and another is getting at the same time. So that will be simple, correct? I promise you fun stuff, but no problem. The another part, how can we transport the big substances through the membrane? They sometimes are too big that, and we cannot just feed them through the channels. We cannot feed them through the pores. There is a way to bring them in or kick them out by the ways of the formation of the vesicles. The vesicles are the small sacs. You can see this, how the vesicle is formed around bacterium here. It's going to engulf it or eat it up. So we gradually build some kind of appendages which will surround and create a vesicle inside. So this structure will be brought in, and again we kind of creating a small bubble with the cell, and of course that requires a lot of energy in order to move the cell membrane, surround it, and again pinch it off and bring it inside of the cell for the future use. If we have to excrete something, the same way we can do that by forming the vesicle, the bubble, combining this with the phospholipid bilayer and opening to outside of the membrane. If you, but like in real life, in the self life, we have substances both solid and we have the liquid substances. It just because of the kind of like state of the substance, we have two parts. We have pinocytosis when it's basically cell drinking, and we have phagocytosis when the cell is eating or engulfing something relatively solid, but the process is the same. The particle, the vesicle is formed, it's fused with the membrane, and either we bring it in or out. It depends on it's endocytosis or exocytosis. And again, endo means we're bringing something inside. Exo means that we are removing something from the cell. There's the schematic representation, and also we have the electron microscope magnification many times, and we have the in purple we have the structures, whatever is being exocytosed here. Well, it's created right there. And it could be anything. It could be the products which are produced by the cell. It could be dead cells, the stem cell, or pretty much you name it. That works for everything. All right. The membrane part, and now we are going to move inside of the cell and look at the organelles. So one of the first organelles which we're going to talk about, that will be our mitochondria. Mitochondria is the part where our cell is producing those ATPs, adenotriphosphate. The majority of them are produced, not all of them, but majority. And we also call it the power plant of the cell. and the presence of these organelles required in the cells which are very active. Especially we can see a lot of them in muscle cells because they're constantly contracting, they're moving the cytoskeleton inside of the muscle cells, and we do use a lot of energy to do that. These mitochondria, they do have, interestingly enough, they do have their own DNAs, they do have their own enzymes and ribosomes. They're almost like a subunit of the cell inside of the cell. If we look closely, they are like jelly bean or bean-shaped structures, oval, and then do have extra double bilayer inside of the membrane inside of it, and they create this invaginations they call the cristae. Why do we have them? Through this membrane, there's actually the cleavage of the chemicals occurring, and that here we created the molecules of ATPs right here. If we look at the membrane of the mitochondria, again, we see the same channel proteins, we see the same kind of enzymes and transport proteins which are occurring in the cellular membrane. So it's like an extra organelle with the membrane there. It's very important, the whole process of adding extra phosphate group to the precursor of ATP, adenosine triphosphate. We put another phosphate group to adenosine diphosphate. That's where it's actually occurring in this membrane of the mitochondrion. But for now, if you just remember that the mitochondria, they're responsible for energy production for the cell, I think, and a basic structure like the mitochondria has cristae and it has the membrane there. I think that will be enough for this class. Just be aware. And mitochondria is a plural. One will be mitochondrion. So, but we have many, so we're kind of very, surely we can say that in the plural, mitochondria. The next, what's going on? A small subunit, it's not matching with my. I also would like to mention the peroxisomes, and this is a small structures which are located inside of the cell. Inside of the cell. Here we have the lysosome, and peroxisome, they are very closely combined, closely related, and just peroxisomes, they have, not like regular lysosomes, they have the hydrogen peroxide, and they are responsible for the oxidation of the toxins. The oxidations mean the destruction of it. Especially, I have to mention the alcohol and this oxidation of the toxins in the liver. It's very important to get rid of the toxic substances in the body, and them being processed in the form, the way which is not very active, and it could be easily excreted through the large intestine, like with the feces, and also kidneys with the urine. What they also do, besides the oxidizing toxin, they also break in fatty acids in the smaller molecules, and these fatty acids could be also used to rebuild to more complex molecules or excreted if it's necessary, or also used for the production of the fuel, ATP production. Ribosomes, another organelle which is composed out of the two parts, large subunit and a small subunit. What do? They are protein builders. Remember, we are talking about the nucleus, which had inside of it the information storage in the shape of DNA, and then RNA will come and grab the necessary pieces to produce blueprints to produce the proteins. And ribosomes, this is the workers, the parts which will actually make these proteins. And they are solid structure, or they are not non-membrane bound organelles. They don't have the hollow part. And where could we find them? We will find them either floating free inside of the cytosol, or they also could be bound to the membranes of the structures, other structures. Either it could be envelope of the nucleus, or it could be also the hollow structures, and they are producing the proteins inside of them, like endoplasmic reticulum. To the parts which are the organelles which are having a solid part, we call it the solid, basically solid non-membrane bound organelles. Or we have membrane bound, that means that we have membrane, they have the relatively hollow space inside. And when talking about membrane bound organelles, that means they containing something, because we have to separate again two liquid parts, intracellular fluid and whatever is inside of the organelle. It's usually the production there, if there's a site of the production or the site of the destruction there. So we don't want these compounds to be mixed with the main cytosol. We have several organelles, where like endoplasmic reticulum is the have these membrane-bound organelle Golgi apparatus, already mentioned peroxisomes, and lysosomes. That's another kind of the storage vesicles. Endoplasmic reticulum surrounding our, so there we have the nucleus, and there's a structure of membrane-bound organelle, a relatively hollow, like kind of labyrinth surrounding the nucleus. And you can think this way: if we have the genetic material stored inside of the nucleus, and the purpose of the cell, for example, produce some kind of proteins for its own needs or be excreted for the needs of the rest of the body, where do we get the information? We get it from the nucleus. And it makes a lot of sense to bring the plant or building structure close to the site of this information. That's why it's surrounding the nucleus. So immediately the RNAs, they don't need to go too far. They send messengers there, and ribosomes, which are embedded inside of the nuclear envelope and also in the walls of the endoplasmic reticulum, and we can see like kind of bumps. That's why it's called rough endoplasmic reticulum. That's where the oil production is going on. And when the production is finished, the end products that gets accumulated within this convoluted labyrinth there, And if it's necessary to excrete it, we easily can create like a moving containers in the shape of the vesicles and either remove it from the cell or move it to another part of the cell. We also have another part to the endoplasmic reticulum called smooth endoplasmic reticulum, and does not contain any ribosomes in it. The Golgi apparatus is very similar in structure to the endoplasmic reticulum, and but the function of it, if we compare it to the endoplasmic reticulum, which stores stuff and pumping it out in the cytosol, or we're doing the detoxification reactions, and also doing some lipid synthesis, the Golgi apparatus, it's mainly it's secreted from the self-exam or it's sent to the lysosomes. What do we do? What do we transport from the endoplasmic reticulum and to the Golgi apparatus? These again produce proteins and lipids. They get sorted there and further transported throughout the cell or even outside of the cell. A little bit of caption here. Again, we get the information from the nucleus, build the proteins inside, then transfer it to the Golgi apparatus, and with the lysosomes, endosomes, is it possible to move it? Yes, it's possible to move it outside of the cell or move it throughout the cell. What do you need to remember? Remember the names, remember the structure, remember, is it bound by the membrane or it's solid, and approximate function. Now we are moving to the cytoskeleton. Cytoskeleton, this again, I would repeat that it's a bunch of like a framework and gives the shape and size of the cell. That's usually made out of the proteins, which are arranged in a certain way. They are fibers or filaments. They are long, elongated, attached either to the organelles or to the cell membrane, because they cannot just float inside. In order to be a part of the skeleton, they have to be attached. And what do? They do provide the framework. Sometimes they associate it with the movement. Again, in order to move something that's exactly like skeleton in the body, we have to attach muscles to the skeleton. And here we have to have this attachment of the cytoskeleton to two parts. And of course, for the movement, it requires energy. So we have to have the mitochondria with the production of ATPs, constant production of ATPs available for this movement. What can we find inside of this cytoskeleton? We can find the actin filaments, and we have intermediate filaments and macrotubules. They are all just different in the size. So we have the smallest one is actin filaments, the thinnest one, then organization into intermediate, something in between, and macrotubules. They actually, what happens is filaments, they are bundled in a spiral to give structure, but at the same time maintain low weight of the construction here. One of the important functions is also the centrosome, which is in participating in the formation of the spindles, which are pulling the genetic materials to the the dividing cell in the equal parts during the mitosis and meiosis. When we go through this first and second stage, we can equally divide everything. Here we can see the microtubules in the pairs, and they are attached, they actually becoming the skeleton for this appendages called cilia. And cilia is like a hair on the top of the cell. Of course, they are facing the outside. It's usually not outside like skin, but inside of the intestinal tract or respiratory tract. And what happens there, they have ability to gently move. And if we move in substance, we can move, for example, the dust particles, the bacteria, whatever mucus on top of the cells out of our body. This is one of the functions there. Nucleus. Again, I mentioned it several times already. Let's look at it a little bit closely. This control center, it directs the activities of all the cellular components. And why do we do that? By the presence of DNA. And of course, DNA, they are not moving out of the nucleus. They're not going anywhere. Only time when the cell is dividing, this DNA is unfurled for a relatively long period of time, then they could be divided evenly, replicated, and then pulled apart. But if for some reason we need to create some certain part to express the gene, the RNA will copy the information, and but DNA always stays inside of the nucleus. As you can understand, it's very precious. What surrounds the nucleus? It's a nuclear envelope, another membrane, and we have the rough endoplasmic reticulum surrounding that. You can see the little bumps. This is our ribosomes embedded there. Again, we are extensively producing the proteins. Nuclear pores are the openings, they connect in the neoplasm or cytoplasm. I cannot say cytoplasm, but the plasma looking structure within the nucleus with the cytoplasm of the cell. In case we need to transport the substances, again, we have to do the nourishment of the inside of the nucleus. We have not and remove the waste products. This is the nuclear pore. Probably everyone familiar with DNA is a double helix there, and it's a normal way it's tightly wound in the shape of the chromosome. The nucleosome is the strand of DNA called around the histone proteins. Again, this is just information for your general general information for you. There will be no questions about it, but this double helix of the DNA even further found in wound up around the histone proteins, and even further it's kind of folded on each other and created a very compact chromosome right there. I will kind of skip this part. You can, I will actually pause the lecture about the protein synthesis and how we go through that. You can look at it in your spare time, refresh your memories from the high school. Okay, here we go. We have to talk about the mitosis versus meiosis and what happening. Okay, at this point, we have two parts. We have the meiosis and mitosis. What is the difference? Can someone tell me what's the difference between the mitosis versus meiosis? Let's wake up and kind of start talking.
Is it
Wait.
meiosis
Yes,
with I think it's sexual reproduction,
yes,
and then I think mitosis is regular cellular reproduction.
yes. What is sexual reproduction? Why do we need to have two organisms to reproduce? Why don't just kind of continue with mitosis?
Because we need more information from two people. So 23 pairs from one person and 23 pairs from another.
Why do we need that?
So because we need more genetic diversity.
Why?
first of all. So, and also to fight against a certain type of disease, because if we were all the same genetically, if we had a disease all of a sudden, we'll just all perish.
Absolutely. You're absolutely right. Remember, the natural selection is what? Survival of what? The most adaptable.
Strongest.
Yes, no, not the strongest, not the smartest, but the ones which could quickly adapt to some kind of situation. And if we kind of look at it very cold-blooded and see, okay, we have very diverse genetic pool because we keep taking these chromosomes from mother and father, and the new generation is every single person is very unique. Even if you have identical twin, you start with the same genetic makeup, but later in life there is events happening to you and mutations, and at the end you are not exactly like your twin. That allow us in a big genetic pool, for example, there always will be even a small percentage which will be immune to, for example, some kind of disease just because of the genetic makeup, because the wide diversity of the genetic information in us. Exactly, that's exactly right, whatever you said. And during the mitosis, however, we have the DNA replicated, and we are not exchanging the information. We're creating pretty much the same cell as original. That's how we can replace the cells which are worn out, which are dead, by the means of creating exactly the same cell in as the mother cell, just creating more than dividing in two. And we have to replicate, and we have to give each cell the almost the same amount, as much as possible, even division. That's how we have our cytoskeleton kicking in and pulling not just genetic information, not just DNA parts, but also the resources like cytoplasm and plasma membrane and organelles. They are equally divided between these two, like inheritance. Like, yeah, you got 50%, you got 50%. Here we go, new start. When we're talking about the cell and mitosis, we have the 4 parts to it. So we have G1 S phase, G2, and then we have M phase. or actually the mitosis phase. During the interphase, which basically the cell from the daughter cell, just newly divided cell, it growing, it's acquiring information, no, not acquiring, it acquiring the resources, it's acquiring more cytosol, it acquires probably the organelles, and it becoming full grown and ready to divide again, depending on its position, depending on requirements of the body. Again, not all the same, all the cells dividing all the time. Just usually there is a certain group of the cells which are precursors to the tissues. They are constantly dividing, and later they are kind of undergoing specialization and maturing and acquiring the properties which working cell needs in order to create the tissue. But we will talk about this part when we do the histology. From now, I just want you to know what is mitosis, what is interphase, what is G1 S phase, synthesis phase, and G2. That is for the lab. We will see the stages of the mitosis, like anaphase, telophase. We certainly do that part. Okay. Sorry, my computer is sticking again. All right, sorry about that. The parts of the cell cycle, again, interphase, the everything was growing, so nucleus. Unfortunately, during the lab, we will not have chance to look at the microscope. If your job requires the usage of the microscope, you probably unfortunately have to learn how to do it on your own, and maybe there will be some training. I cannot bring you to the class, but I will provide you with some movies, some videos. You can see they actually maybe better than if you could see it with your own eyes under the microscope, because they are using the electronic microscope and it's much better resolution. But if you look under the microscope and see the cell in the stage of the interphase, you will clearly see the nucleus. Sometimes when you're looking under the microscope, the first thing what you see is a nucleus. Sometimes you cannot even see the cytoplasm. Definitely you cannot see the plasma membrane. Maybe if the microscope is very good and the cell depends on, again, on the cell, you might see some mitochondria, but the nucleus, that's pretty much what you can see, and some cytoplasm, because of the coloring, because of the dyes which I used in order to make the cells more visible. In normal conditions, without dyeing the cells, they are very transparent, translucent, so you in normal situation you cannot see it very well. But here, for studying purposes, we did some schematic representation and corresponding slides. This is, I do believe this is the magnification, the electronic microscope slides you see here. But anyhow, the interphase, that's where you can see the nucleus, because the nuclear envelope is still intact. The cell is not doing much division. It's just getting ready. And chromosomes, the chromosome material, it's all bound kind of like a ball of yarn. You cannot distinguish each chromosomes from each other because they are not prepared to be divided. They are not separated. As soon as cell says, okay, we had enough of the division accumulation, it's time to divide it, then cell is going into the mitosis, and mitosis itself has stages. I wish I can make it bigger for you, but stages that starts with the prophase, when the each individual chromosome has two sister chromatid, and it's like this bow tie representation, and nucleolus itself start to disintegrate because it's going to divide. We cannot contain this chromosomal material inside of the nucleus anymore. We're going to stretch it in one linear. Yes.
Interrupt Janelle. She got kicked out of the Zoom and she's asking if you can add her.
Sure.
Thank you.
Thank you. Thank you for watching over each other. So when we are going to the next phase, metaphase, the genetic material, genetic material is getting stretched along the equator of the cell. The nucleus envelope is not existing anymore, and we have the spindles, the fibers from the cytoskeleton attached to each chromatid, and it's getting ready to pull it apart to opposite poles of the cell. When you look under the microscope, you of course don't see all this nice drawing, but you see this like darker line of the genetic material in the middle right there. It's almost looking like a starburst or fireworks on each side. And this is the center of it is the central pairs right there. Stage of the anaphase is the next one. Yes, next slide. Oh, okay. The next stage will be the anaphase, when the chromosomes, they are already separated and they're in the process of being pulled to the opposite sides, but we cannot see the formation of the new nucleus yet. They're still attached to the spindles, but hopefully if everything went right, we have the equal amount of the chromosome materials on each side. The next stage, which the cell is going to call telophase, and the telophase, we already see the two daughter cells being formed. It is not just the all genetic material start already divided and started to be separate, separated from the rest of the cytosol by the envelope. But also we can see the cleavage of the cytoplasm, and cleavage means the furrows forming and two kind of bubbles from one. This is the stage of the telophase, and under the microscope you can see almost two cells almost separate there. We have the deep furrows, that cleavage right here, but they still have some kind of connection here. They're not completely separated. And when everything is done, we have two brand new daughter cells which are going through the interphase again and accumulate the material. And when they are ready to divide, they will go through mitosis again and again as needed. Just what's happening there. Why do I have TV? what we can do, we are going to take a short break. Let's say how many? 5 minutes will be enough for you to do whatever you need to do, stretch, go outside, get a drink of water, use the bathroom. And after the 5 minutes, we will continue with our breakout rooms. You will be divided again in 7 groups. You will pull the question which is corresponding with the number of your group, discuss it for a few minutes, write the best answer. I would suggest you to do the collaboration, do the same answer. It will be probably save some time for you and your friends, and then we'll get together again. You will do the presentation. They will get 1 speaker or 2 speakers. It's up to you. Present your part, and we go one by one. And after that, again, short break, and we continue with the next lecture. Do you have any questions?
No.
No? Okay. Short break. I'm immediately putting you into your break room, so you will, from your break, you'll just join your friends in your group and start working on assigned questions. If you have any kind of questions for me, I will pop in and out from the discussion groups, but also there is a feature on the Zoom that you can ask me for help, like raise your hand or something. There is somewhere on the bottom you can see, ask for help. Okay, I'll see you in the break rooms.